In this video we will discuss about how to enhance cell surface reaction by macromolecular Crowder. And one of the application of this enhancing the cell surface reaction is about this blood scarcity. So how one can differentiate blood group A and blood group B is through the presence of this carbohydrate structures on this blood group O glycans. So and these carbohydrate structures are different. That is, one of them is alpha galactosidase and another one is N-acetyl glycosamine. So when blood group A interact with blood group B, it will cause antigenic reactions. However, blood group O does not have those extra carbohydrate structures. So that's why blood group O can be considered as a universal donor. That blood group O can be donated either to the blood group person with the blood group A or person with the blood group B. So by enhancing the cell surface reactions, what the researchers tries to do is that they want to convert either blood group A or blood group B into the blood group O by consuming less enzymes. And if those enzymes can be removed, then those treated blood group A and blood group O B can be used for the patients where blood supply is required, because once they are converted from blood group A or blood group B to blood group O, they can be donated to any persons, as these blood group O persons are universal donor. So the objective is that the objective of their study, or the hypothesis of their study, is that they have used some macromolecular Crowders, and some of the results are shown in these other panels, such as in the panel B. They have shown that when they use this EMG 109 without Dextran is one of the macromolecular Crowder. When they use Dextran, they can see a 100-fold increase in the reactions. And this y-axis, this agglutinations represents the enzymatic reactions, and more the agglutination time indicates the more enzymatic reactions occurs. So without any macromolecular presence, they can see a very low enzyme activity, and that too at high enzyme concentrations, whereas with the Dextran they can see high enzyme activity at low enzyme concentrations. And same things they have observed for the other enzymes, such as BFG11OA1. Here they have seen the increase of 440 folds, and for CpGH98 they have seen an increase of 10-fold. And for this other enzyme, that is sp3gh98, they have seen an increase of 80-fold, and for sp3gh98 CD, sp3gh98 CD, they have seen an increase of 40-fold. So it depends on enzyme to enzyme, but the common things about these results is that the macromolecular Crowder will cause enhance in the enzymatic reactions at cell surface. That made it possible physically or practically or economically possible to convert this blood group A and blood group B to blood group O. And they have also analyzed their final treated blood group O for the contaminations of these enzymes, and they have tested using the FDA approved test, and they are able to find out very low enzyme concentrations are present, that is in nanomolar. That is been accepted according to the FDA guidelines, according to the papers that has been, according to the supplemental data presented in the figure. They do not specifically mention that those values are approved are within the limit of FDA, but those values are very low, and with other few modifications we can achieve they can achieve or one can achieve the conditions where these blood group A and blood group B enzymatic treated cells which are converted into the blood group O are being used as a regular blood group O, like a like are being used as a regular blood group O to any patients who needs a blood requirement. So I hope you understood their idea of enhancing the cell surface reactions, and one of their application is to make availability of blood group O to any patients regardless of their own blood group. That's all for this video. Thank you for your time. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description.